Okay, it is now 5:31, so we're going to go ahead and get started. Mahalo, everybody, for joining us tonight. My name is Sarah, and I will be the MC. This month, we are joined by Dr. Mary Hagedorn of the Smithsonian Institute. And tonight, her talk is entitled Saving Coral Reefs, the Ocean's First Fertility Clinic. And in the race to preserve rapidly declining coral populations, Mary and her team use cryopreservation to safeguard the genetic material of as many species as possible in biorepositories and institutions around the world. Tonight, Mary will share her work applying human reproductive techniques like freezing sperm to coral to develop a new tool for preservation and diversification. And of course, tonight's speaker series is it's a monthly talk that we give the first Wednesday, typically of every month at 5:30 p.m. via Zoom. And we would like to mahalo the County of Maui as one of our partners in being able to bring this talk to you every single month. And as a quick side note, if you are joining us, but you're not on our mailing list or you haven't signed up for our Reef and Brief, please feel free to go to do so. We send updates about our talks, our monthly talks, updates on our work, and any volunteer events that we might have. So if you like tonight's talk and you want to be with us next month, make sure you get on that mailing list and you'll be notified when registration goes out. And that link will also be in the chat. So if you are new to Maui Nui Marine Resource Council, we are a 501c3 nonprofit, and we work for clean ocean water, healthy coral reefs, and abundant native fish since 2007. And we believe that healthy coral reefs mean healthy communities and that every living thing on our island, we're all connected, can't get away from it. And so we protect the reefs by focusing our assessment, monitoring, and restoration efforts from Maka to Makai, basically working from the mountains to the ocean. So this means when we work Maka to Makai, our work takes place all over the island. It takes place on land as well as in the ocean. And throughout the year, we do things like testing water quality at over 41 now, actually, locations around the island. We regularly monitor local fish and coral reef populations. And we also provide training to property owners and landscaping professionals on how to use natural alternatives to toxic herbicides and pesticides. And speaking of which, we are very, very excited to announce that we have just launched Hawaii's very first reef-friendly landscaping certification course just last week. So it's very new. This course is designed to help landscaping professionals, property managers, and agricultural businesses to adopt natural land care practices that are just better for plant life and better for our reefs. So this course features a 10-subject curriculum, which is taught by local industry experts, and they'll help landscapers to see a host of benefits, including improved plant appearance and health, and of course, experiencing savings costs on long-term care and maintenance, among other benefits. So a lot of great things about it. It's not just about the coral reefs, it's about a thriving industry as well. And so to celebrate this launch of our course, again, we just launched it last week, we're offering the first 10 students 50% off of the course fee. So you can scan this QR code that you see here on the screen, or we also have a link in the chat. And we'll have it at the end of the presentation as well. Enrollment will end on May 21st, and the course will start the very next day. So if this is something that if you're a professional or you would like to see your provider provide these services, feel free to let them know that it's available, and we hope to see you guys in the course. So if you love MNMRC, we love having our supporters. You can make a donation to us at myreef.org, but there's also a really special opportunity coming up. Ku'ia Estate Chocolate will be having a VIP tour of their factory this Sunday, May 5th. So it's not quite Mother's Day, it's the week before Mother's Day, but all the proceeds from that special tour this Sunday will be going to MNMRC, will be going to help protect the reefs. So if you're looking for a Mother's Day gift or you just love chocolate, and let me tell you, like I gobbled up a whole tin of their stuff already. This is a great way to have a unique gift or a unique experience and to also do some good for our reefs at the same time. So the link to sign up for that is also in the chat, and we hope you guys enjoy it. Let us know how it goes if you do book a spot. So, getting to our talk, a few notes before we introduce our speaker. We will have a question and answer portion at the end. So please submit all of your questions using the Q&A button on the lower edge of your Zoom screen to do that. If you're joining us on Facebook, you can submit your questions as a comment and they'll be relayed to me, and we will, like I mentioned, we'll talk about it at the end of the presentation. We're also being recorded and live streamed. So if you have to jump off or you know a friend that might enjoy watching the recording, this will be available on our YouTube channel in a few days. So just wanted to let you guys know that. Okay, so finally, for our speaker, Dr. Mary Hagedorn received her PhD in marine biology from Scripps Institution of Oceanography and has worked in aquatic ecosystems around the world, from the Amazon to Africa. She's taught many university level classes, lectures frequently to lay audiences, and she maintains an active laboratory with graduate students and postdocs, and is a successful researcher and active grant writer. Oh, shout out for that. Dr. Hagedorn has created the field of coral cryopreservation and founded biorepositories around the world for coral species. Today, she is the director of the Reef Recovery Initiative, a global coral conservation program. So, Mary, thank you so much for being with us tonight. And the floor is yours. Please take it away.
Thank you, Sarah, and thank you, Jill. It's a real pleasure to be here with you all. And so I'm going to tell you about the work that I've been doing really over the past 20 years. And it's not just me, it's many students and colleagues around the world. So this has really been sort of a very collaborative effort. So I'm going to start sharing and get this going. All right, there we go. Right. And so, I couch my work in terms of, sort of fertility clinics because we use many of the same techniques that people on this call may be familiar with because friends or relatives or themselves have, engaged in some aspect of fertility clinics. And it has to do with, freezing, or we call it cryopreserving, where the cells are in a frozen but alive state, and they can stay in that state for a day or 10 years or even 100 years. So it's putting cells and or tissues into suspended animation and saving them for the future or for tomorrow, whatever a person wants to use. So that's what we've been doing for coral reefs over the past 20 years. And as Sarah mentioned, I'm from the Smithsonian Institution. It's largely based in Washington, D.C., but we have institutions around the world. And I work at the Hawaii here on the right. I work in Kaneohe Bay. This is Kaneohe Bay and on the island of Oahu. And this is the Hawaii Institute of Marine Biology. So my laboratory is here, and you can see we're completely surrounded by coral reefs. There's about 50 patch reefs in Kaneohe Bay and many fringing reefs, and we do have a berry reef. So we have all three types of reefs in Kaneohe Bay. This is my lab team, and you're going to hear about most of their work today. We have interns, and I'll even just point out Mari Quinn, who will be graduating from the University of Hawaii in 2 weeks, and she started in my lab in 8th grade. So it's really nice to see her go from junior high school to all the way to graduating from college. Okay, so many of you are very familiar with the idea of climate change, and you know that it's altering our terrestrial and marine ecosystems and threatening all life on our planet. Droughts are far more frequent. Fires on the west coast of the U.S. and sadly in Maui, floods and extinctions. And so today we're going to focus largely on coral reefs. Many of you know that coral reefs are animals, but that's probably one of the most important take-homes for today. They are animals. They are largely located in the tropics. They are one of the oldest ecosystems on Earth, and they support 1/4 of all marine life. They help produce oxygen on our planet. They don't do it directly, but they're in the chain. Their presence protects our homes and cities by breaking the energy from damaging waves, and they create livelihoods around the globe estimated at 10 trillion US dollars per year annually. So that's a really important suite of characteristics about why we really should care about coral reefs. So if you've never seen coral reproduce, it happens a couple of days a year for each species. And coral are hermaphrodites, and that means they produce both male and female gametes, so sperm and eggs. And so I'm just going to show you a little video of coral spawning so you can see it if you've never seen it offshore on Maui. This is the egg sperm bundle. It's huge. And it's composed of lots of eggs and sperm, and they float to the surface. You can see them here. They'll float to the surface, and then the eggs have to be fertilized. Here you can see a bundle sort of breaking up, have to be fertilized by a nearby neighbor. So corals generally do not self-fertilize largely. I mean, some small percentage can, but generally they don't self-fertilize. And they have different kinds of some corals are sequential hermaphrodites, meaning sometimes they're males and then they can turn into females. It's really interesting. And this is one that does it. It's the mushroom coral. We work on it on Kaneohe Bay a lot. And these are brain corals. They're just always very, very cool and beautiful. In fact, we're going to have our first coral spawn in the next 2 weeks in Kaneohe Bay. All right. So as Sarah mentioned, we're using human fertility techniques to save coral reefs. And worldwide, we have created a bank of coral sperm, and the stars show you where we've collected a lot of the sperm, myself and my colleagues. And here's, I mean, I don't want you to read this list, but you can see that it's from all over the world. And we actually have a training, we have a link to these list of species that we've cryopreserved. So the conservation benefit when you cryopreserve sperm, and this is my former technician, Jen Carter, is that you can use that sperm to help diversify shrinking populations. And you'll see an example of that coming up shortly. In our hands, it's at a very advanced stage of development. We know how to do it. We can train people to do it. We have training videos. We make our own equipment. We can show people how to make their equipment. It's very simple to do, but it's limited because, as I mentioned, some corals only spawn a couple days a year, and so it's very restricted as to when you can use it. And we have used frozen sperm, and we've thawed it, and we can make new coral. And so if you look at this image here, it's a really beautiful picture of a Acropora clethrata. And here's its skeleton forming, and then its tentacles. There's 3 little polyps, and then the brown specks are the symbionts that the corals take up. And the symbionts give the coral color, and they also help feed them. So they'll be important as we go along. So we used our frozen sperm in a trial called assisted gene flow, and it was an international collaboration. But assisted gene flow is just a fancy way to say selective breeding. We've been doing selective breeding on this planet for thousands of years. It's called agriculture, but scientists have to sort of get fancy words, and it actually has caused problems, I think, in my mind, that using that terminology rather than just selective breeding. So what we did is, this was done in the Caribbean because I had cryopreserved coral sperm from the Elkhorn coral. It's right here. This is the Elkhorn coral. These are massive corals. This cluster is probably as big as a VW bus. I mean, if you know what that looks like, it's huge, and these corals form the bulk work of support and protection for the Caribbean and Caribbean islands. And in Florida now, they are in really bad shape, especially after the last summer where some of the water temperatures went above 100 degrees Fahrenheit. So what we did is we started collecting and cryopreserving coral sperm from this species in Florida. We also did it in Puerto Rico and then Curacao. And then we crossed these with fresh Curacao eggs. We cannot cryopreserve coral eggs. And so we made all these different crosses. Now, these are not genetic crosses. these are from the same species. We just did a long distance matchmaking with them. And then what we did is we took the larvae that we got and we sent it to Florida, and it was reared in Mote Marine Lab in the Keys and the Florida Aquarium. And they did an amazing job with rearing them. And here are some of the larvae that we produce. It's just gorgeous. But what was really interesting is that we produced over 600 unique crosses. They're not hybrids of Elkhorn coral. That means 600 unique genotypes for this coral. And they're in a tank in at Mote Marine Lab. And the reason they're in a tank is that the Florida Fish and Wildlife prohibited in water testing of our assisted gene flow genotypes. So as I said, they remain in captivity. Today, only 7 founder genotypes now exist for the Elkhorn coral on the entire Florida reef tract. 7. That's unthinkable. I mean, 20 years ago, there were probably tens of thousands. We have 150 that were brought into captivity last summer, but the rest of them died pretty much in the water. And we have 600 still in a tank waiting to, go out and hopefully help diversify the population. So one of the things that we like to do is we like to think about lab to reef. And our conservation goals really is to democratize our science globally through capacity building. And we do that by having training videos and trying to help people who want to use this technology and make it very simple, make it inexpensive, and give them the tools to, to do it themselves. And if they can't, they can always contact us. And I have my email at the end of this talk if someone has questions and want to contact me and ask those questions further. So we're creating biorepositories around the world for coral. We've just, this is a new one that's at the Smithsonian. These are large doors, and they can hold thousands of samples in them. So there's this small room can maintain many thousands of samples. But we're increasing the capacity of at USDA at the animal germplasm repository. We're building repositories in the Caribbean, and I helped build a repository in Australia. So we're expanding our network around the world to be able to hold this material as we start to cryopreserve it. So in addition to sperm, we can also cryopreserve coral embryos. Now, when I first started this project, I wanted to just start on embryos because the conservation benefit is huge for embryos, because first of all, you have a sexual assortment because you've got a male from one colony and a female from another. So you have a new genetic assortment that may have some really advantageous changes or adaptations to the environment. And so that's always important. And if you can freeze the coral larvae, this is a coral larvae, it will prevent extinctions. However, the coral larvae are very chill sensitive. And when I first started this, I would start cryopreserving them, or what I thought was cryopreserving them, and inevitably they'd end up like this ball of fur. And after about 4 years, I stopped because I realized I needed new technology, and I wasn't going to be able to move forward with these studies with the tools that I had in hand. And so in 2016, we held a laser workshop at the Hawaii Institute of Marine Biology, and we partnered with engineers at the University of Minnesota, and John Bischoff and his team brought laser warming to Hawaii, and we had applied it previously to fish embryos, and then we quickly applied it to coral. And the 2 people that took this work and did it, along with these other colleagues, were John Daly and Nick Sahavich. And here's John shooting lasers around the room. It's really not. They're just, I think they're just laser pens or something. I'm not really sure. But what we did is we used, the laser could warm the material really fast. And we used gold nanoparticles in conjunction with these lasers. And the lasers cannot really warm biological material. They actually at this frequency pass through biological material, but they will warm gold particles. And so we use the gold as radiators basically. So we hit the material with this laser, it warmed the material, but it really warmed the gold, and the gold radiated heat into the material and then warmed it. I'll show you a little video of how we do this. So this is Nick's demo. So we use these little paddles, we call them cryotops, and we're going to put a tiny little droplet of material that's got larvae on it, and then you can see there's this gold color that has the gold particles. And we're going to take that sort of stick and with the droplet on it and put it in liquid nitrogen. Now it's completely frozen, but with no ice. And that's really important. We call it vitrification. And if you can get rid of the ice, then you can get rid of the damage. Now, when we want to warm it, we bring it up and the laser hits it, and we've warmed the larvae. So when John and Nick did this, it took them all day to produce about 100 larvae post-thaw. So they freeze them and then thaw them, and we got about 100 larvae in a day, which was great because it was, we could go from no survival to having almost 30 to 40% survival in a day, which was really great. But this was really just a proof of concept, not a practical tool for restoration, because it's very hard to train people in how to do lasers. And, it's just very complicated, and it's more expensive than we wanted. So we wanted something that would be simpler and easier to apply, but we knew the principles and we knew what we had to go for. And we worked with engineers to help us make it simpler and easier and cheaper. And so we ended up using this thing called mesh technology. And you can imagine that this is kind of like the mesh in your Windows screens. It's not quite, because it's metal, it's stainless steel. And all these little specks here are larvae, coral larvae. And we've frozen them and thawed them using the mesh technology. And we're able to do hundreds in just a few minutes. And you'll see that after 2 hours or so, after we've frozen them and thawed them, they just start swimming around. And so this is a really amazing and simple technique to do larval cryopreservation. Now, it doesn't work on all the coral larvae, because as the coral larvae get much larger, there's a limit in terms of the volume that you can freeze with this technology. And but we're working on ways to try and make it better and make it so it can apply to all the coral larvae. And some of the really big larvae are the ones from the Acropora corals from the Great Barrier Reef and other places that are the reef building corals. So as I mentioned, you're very familiar with climate change, and in Kaneohe Bay, we had 2 really bad bleaching events in 2014 and 2015. And when we talk about bleaching, what we mean is that the coral has turned bone white. It's not dead, but what it's happened is that during the warming event, the symbionts that live inside the tissue of a coral, they give it its color, and these two still have their symbionts and still have their color, but they also feed the coral. They take sunlight and they turn it photosynthetically into sugars. And when the symbionts get stressed, then the symbionts leave. They break the symbiosis and they flee. So what you're seeing is coral tissue, which is transparent, and you're looking right at the skeleton, which is bone white. Now, if the corals do not get their symbionts back in about 10 to 12 weeks, they starve to death. And but as I'm sure you know, any animal that has starved for even 6 or 8 weeks, they've used up a lot of their fat reserves. They're physiologically not in very good shape. And so climate change and the warming that comes with climate change is negatively affecting reproduction. And corals can reproduce after they've bleached, but oftentimes the larvae don't live, or they don't settle. And we have a very hard time cryopreserving them. If you've had the pleasure of going to a fertility clinic, you'll know that you have to have a certain level of physiological robustness to the sperm or the eggs or the embryos that you are going to have cryopreserved. So it's a much higher bar than just, having coral sperm and eggs get together and produce natural reproduction. So in order to try and expand our efforts and help ourselves go faster, we partnered with more engineers at UC Berkeley, and we started using this technique called isochoric vitrification. And I'm going to explain it a little bit in a second, but we use the coral in the bay called Parietes compressa, very, very robust and tough coral. And what we do is we take small pieces about like this size, and then we cut them into even smaller pieces. All these little squares are tiny little pieces of coral. They're about the size of your thumbnail. We glue them to the plate, and then we let them grow out so that they start to heal and sheet. Then we take those growing, we call them microfragments, and we can test them for cryopreservation. And one of the techniques that we're using is this process called isochoric vitrification. And basically, I want you to imagine that you've actually kind of done this sort of experiment a bit in your home, probably, or one of your kids or whatever. If you take anything that's in a can, it could be soda, it could be a beer, it could be anything, and you put it in the freezer to cool it down. And if you forget about it, you'll notice the can expands, and sometimes it even bursts. And that's because water takes up less volume in a can than when water freezes. So the can expands because the water in the frozen state has expanded. Now, if you had a very rigid structure like we have here, this is an aluminum structure. And then we have cryoprotectant here. And when we freeze something, we want to get, it's kind of like when you put antifreeze in your car to protect it from getting too warm or too cold. And that cryoprotectant or the antifreeze is very similar to the cryoprotectants you use on tissue. And you want to get that cryoprotectant in and water out. And this is a very viscous solution, kind of like syrup, and it has very special properties where it doesn't form ice. It just goes from the liquid phase into a frozen glass, and the coral fragment just sits here. And as long as there's no ice in the solution, then the coral lives. And the thing that's unique about this is if ice does try to form, then the rigidity of the walls, these aluminum walls push back and stop ice from forming. So it's this isochoric vitrification process that we've been using. And what's really cool about it is that we can freeze coral for 365 days of the year. We can go out every day and try and freeze coral. Now, this is still very much an R&D. We've had a lot of successes, but we are not ready to take it out into the field just yet. But it is coming, and it'll come within the next 6 months. And ideally, what we want to do is form a conservation corps and go around the world and train people to do this and use it, and to quickly form their own banks with coral fragments. So in addition to these kinds of sort of cryopreserved banks where you have tissue that's frozen and alive, we're also sort of using the power of existing networks. And we formed this thing called the Coral Biobank Alliance. And it's a collaborative network to preserve all 1,000 species of coral in captivity. And the goal is to bring them in and to keep them alive for the next 100 years if we have to. To date, we have over 260 species in captivity. And basically, we want to store the genetic diversity and biodiversity of corals. We want to standardize collection and data management across the globe, and train professionals and build capacity. We have some founding partners such as Smithsonian and Taronga in Australia, the Great Barrier Legacy in Australia, the World Coral Conservatory, which is throughout Europe, NOAA, which is throughout the US, and then AZA, Coral Safe are some of our partners. So the goal then over time is in the next 50 plus years, we have all these aquaria that have brought corals into captivity, and they're maintaining them. and we bring them in and we keep them in live culture. We can try and cryopreserve them and cryopreserve their symbionts and their microbiome, which is on the surface of the coral. Like we have a microbiome in our gut. It's basically bacteria that keeps them healthy. And we can also take tiny pieces for genetic identification. And then over time, we can take these coral pieces, these coral colonies that we brought in, we can grow them up, and there's ways to have coral breed in captivity, and these facilities exist all over the world now, and corals are breeding in captivity. So you can do selective breeding or however you want to do it, and then you can take the embryos or the egg sperm bundles and make embryos that can go out for reef restoration or whatever. We could freeze them. We can do whatever we want, but this is the plan for these corals when they come into captivity. We can do a lot with them and really do a lot to maintain the genetic diversity and biodiversity of coral. So I'm going to just talk about one last thing before we sort of pause and take some questions. Many of you may have heard of a place called the Svalbard Seed Vault. It's a global biorepository for all the world's seeds. And what's amazing is that this is a picture of it, and it's dug into the permafrost in the Arctic Circle of Norway. And the level that it's dug into, the temperature in the seed vault is at minus 18 degrees centigrade, which is perfect for maintaining seeds. So it's called a passive biorepository in that you don't have to add any energy. You know, the seeds can sit at this temperature for tens of hundreds of years, and basically people don't have to do anything to the seeds. And if they're needed to help reconstitute agricultural or terrestrial ecosystems, they're there for that purpose. We would like to do the same thing for wildlife species, but there's no place on Earth that is cold enough to hold cryopreserved material, because we hold cryopreserved material at minus 196 degrees centigrade, which is way colder than any place on Earth. And so we could hold it in space and have a lot of this, these material in sort of a wildlife biorepository as a satellite, but then you expose the material to a lot of radiation. But if you go to the moon, you can go to the southern pole of the moon, and you can dig the material into the it's called regolith. It's the soil of the moon, and that if you go down about 6 feet, you block most of the radiation, the solar and cosmic radiation. And these are my colleagues that are helping us work on this lunar biorepository. It's really just a concept right now, where we do have some materials that we've collected here in Hawaii. And this is the area of the moon that we'll probably try and store on it. It's at the South Pole. And this blue area, means that it has consistent temperatures throughout the day and throughout the seasons at approximately 196 degrees centigrade or colder. So this is a really great place to have a passive biorepository. And some of the sorts of groups that we might bring are ecological engineers, such as parrotfish or coral or beavers, whatever, pollinators, such as bees, extreme environment fauna, primary producers, such as bacteria and other things that may be used for terraforming outer worlds, fishes, threatened or endangered species, organisms that are critical for spaceflight or terraformation, ancestral wild relatives of important agricultural species, and species of cultural importance. This is just a beginning list. I'm sure things will get added to it as we go along. There's also a whole list of plant species that cannot be held as seeds, that have to be cryopreserved. A lot of them are plants that live here in the tropics, such as bananas and mangoes and things like that. They have to be cryopreserved, so they would be included along with probably other seeds in this biorepository. And we've started, this is the starry goby, and we collected them in Kaneohe Bay. And basically we decided what we would do for this biorepository is we would collect skin samples for all the animals, because we don't know how to cryopreserve the sperm or the embryos from a lot of wildlife species. And so what we do know how to do is we know how to cryopreserve a thing called fibroblast cells. And you get fibroblast cells by taking skin, or if, in this case, a fin from a fish. And we cut the fin off from the gobies and we put them in, we cryopreserve the fin. We took a DNA sample and we put those in the Natural History Museum in Washington. They're now waiting. And when we get some money for this next phase, we will take those fins and we will create fibroblast cells. And these are the little black dots you see here. And those are common to almost all skin species of wildlife, and humans included. And it's commonly done when you clone something, you make fibroblast cells. And the fibroblast cells can be turned into germ cells, like can be made to produce new animals. And this is how they did a lot of the cloning. So we need to test the fibroblast cells on Earth to radiation, the kind of radiation that they might get going to the moon and perhaps coming back from the moon. And we'll hopefully do some of that in the International Space Station, which we see here. But I just found out yesterday the space station is coming down in 2029 or 2030. It's going to come out of low Earth orbit and be destroyed. So we will have a lot of time to do this. But the goal is to see how well these cells do. Do they get damaged? Is their DNA damaged? Can they form, new fibroblast cells? There's a lot of testing we have to do before we send them to the moon. I think with that, I'll stop and see if there's any questions, and to thank you and my funders and all of the people in my lab and collaborators who contributed to this work. And this is my email if anyone would like to contact me. So thank you very much for your attention, and I will start taking questions if you have them.
Thank you so much, Mary. Just a quick reminder that if you want to submit questions, please use the little Q&A button at the bottom ledge of the screen. So Mary, one question is, so who I think you briefly mentioned this, like that there might be intent to, create a process that is less expensive, more easily replicated. And so just, when that is made available, like who would be able to replicate your process if and when it's ready for wider distribution? Would it be, just scientists? Would it be universities? Or would the vision even be for like an environmental organization to be able to replicate and help do this work? Like what's the vision for that?
So each phase, so I started out with sperm. That phase is ready to rock and roll, and it's on our web. We have training courses, and we've had people just read the training, go through the training, and did it themselves. It was incredible.
Oh, wow.
So you can build, I mean, there's ways to build the cryo freezers because they're all 3D printed, very inexpensive. You could get up and started, if you had coral spawning in your backyard, you could get up and be able to do this with like $100. So it's not, that piece is not terribly expensive. The coral larvae in the next year or so, we hope to have that at the same phase where it's going to be very inexpensive. And we will be training coral professionals to do that, and managers, any professional that really wants to do it. We go to this meeting called Reef Futures, and it is a meeting of coral scientists and coral managers and coral professionals of every stripe. And really anyone who wants to participate can, as long as we all try and agree on standard methods. And I think that's really important, and that people know where the material should go and how it should be, how they use standard ways of doing it. So it's a bit like trying to, create a cookbook and have everyone, use the same recipes, so you get the same good outcome. And so for the coral fragments, the same thing will happen. It will be inexpensive. We will be able to train people really easily to do it.
Awesome. Another question, do the sperm that float to the surface have natural attraction to their reproductive match to form an embryo? How do they find each other?
Really good question. Well, I mean, so here's something to think about. When the Great Barrier Reef spawns, so there's about 400 species of corals on the Great Barrier Reef, and they vie for time. There's only like a week that all those 400 are spawning, and so they start spawning generally around 6 o'clock, and so they're time sharing. Almost every half hour, another species is going off. And so there's a lot of material that's released into the water, and they, as long as you have enough, coral colonies close to each other, they just naturally form, the sperm can swim to them and or be drifted to them from the currents. But what we've seen in the Florida Keys is that there are not there on each key, there's only one colony really, even though there may be others around, they're clones of each other. They cannot self-fertilize, and the other individuals on another key about 20 miles south or north, as the case may be, and they can't interact. So when that starts happening, then you get breakdown of reproduction.
Another question, what is the overall scale of the task? And perhaps that means, the whole process. Has there been like a defined long-term plan? Is that something that's still in the works?
Another really good question. So this year we are going to come up with a sort of global plan for all the different aspects of of sort of coral biobanking, whether it's bringing corals into captivity or whether we're actually cryopreserving them. We're also going to be doing things like just taking fragments and freezing those, because you can get a lot of genetic information from those. And so we have various levels that will be part of this biobank, and we will have a global strategy for some of the places that have the greatest risks and how we might be able to mitigate those risks. And what are the resources that we're going to need to get those to happen effectively? Because I mean, to a large extent, in my mind, and everyone has a different timeline, right? For cryopreservation and for banking, it's a much more restricted timeline. We probably have 10 years left in our oceans, which is not much.
Wow, that's a sobering statistic.
Very sobering. I mean, that doesn't mean
Yeah.
corals are going to disappear by any means, but
Right.
we won't really have the capacity to do much beyond that, because the corals either won't be in either it won't be in good shape, or they'll be so few and far spread apart. They'll be really hard to work with.
So actually, this question is kind of similar to the ending part of that answer. At the rate that coral is currently being cryopreserved, do you believe you'll reach the goal of preserving all 1,000 species? Is there a time frame that could be completed by? Or maybe now after hearing that, what do you think is a realistic goal for preservation within, I guess, the next 10 years?
I think we'll do it. There's the community has such a huge fire underneath them. And will everything be perfect? No, but we will get to some of the more important areas of the world. And whether we just have their DNA and their microbiome cryopreserved, or whether we have, fragments cryopreserved, or whether we brought colonies into captivity, it'll vary in each place, because like there's some places, such as Indonesia, where we cannot collect because they've signed the Nagoya Protocol. And that means you cannot take any genetic material out of that country if they're a signatory of Nagoya. And which is great, because that stops biopiracy for that country, but they
Mhm.
didn't put any sort of codicil in there for conservation or scientific studies. And so it makes it very difficult. And
Um,
so we have to look at ways where we can perhaps work on the edges of Indonesia and try and get some of that genetic diversity and biodiversity by working in other places, such as Borneo, or perhaps East Timor, or who knows. But there are some areas that are so important, but we just won't, and there isn't enough time to change Nagoya. There's a lot of scientists who tried working at the UN to try and get Nagoya altered so that there could be some, some codicils for science. And there's just too much, it just takes too long.
Yeah. Yeah. Oh my goodness. How do you know when a spawning event will take place? Do you go out to certain events and like not others? Is there like a criteria that kind of triggers you conducting the process that you're talking about?
Another really good question. So here in Hawaii, I know every single coral spawn that's going to happen this summer, and I know exactly what day and I know exactly what time because the corals are so dependable. And in fact, we have people coming from Washington, and the first spawn will happen on May 21st at 7 a.m. And and so um we have it just mapped out throughout the summer. But that's not true of many places in the world. You really have to know what species are there and when they're going to spawn. And sometimes they're not that reliable. It depends on what's happened to them. Like if there's been a typhoon, or if there's just so many things that can affect, the coral. But like on the Great Barrier Reef, I mean, there's so many species, and they spawn in such massive quantities that you usually can get something in Australia. And throughout the Caribbean, we pretty much know when a lot of those corals will spawn as well. It's places where people really haven't taken the time to, like in Kaneohe Bay, when we wanted to add a new species that we didn't know when they spawned. It took a whole summer of doing actually
Mm.
a whole year of histological collection, so we could actually look at the coral and see when their ovaries and their testes were developing, so that we could then say, okay, that's the month that we have to look. And the next year we looked at that month, and we just had to watch kind of 24 hours during that
Wow.
time to figure out when they spawn.
Hey, I had no idea. That's very interesting. Let's see. I think that was all. Just checking all the different places, making sure. Yeah, I think that was the last question that was submitted. But like Mary mentioned, you can reach out any other questions via email. And then trust me, people, if you just Google her, like she comes up instantly. So she's pretty well known.
For the good or the bad.
So if you have any other questions, please feel free to reach out. Thank you, Mary, for being so gracious with your time tonight.
Of course, it's been a real
Thank you.
pleasure.
Thank you
Good
so
luck
much.
with your good luck with your conservation efforts on Maui. It's so needed. Thanks
Oh
so
my
much,
goodness.
everyone. Thanks
Please,
for your
please let,
time.
please let us know when you're on Maui. We'd love to
Okay.
we'd love to hang out.
Okay. That sounds perfect.
So
See you.
thank you everybody tonight. Thank you to Dr. Hagedorn for being with us. And of course, once again, mahalo to the County of Maui for being a partner in this speaker series. And of course, thank you to everybody who is watching. We do this for you. So thank you for being here. And yes, like you just saw, our next speaker for June will be Evelyn Tickle, the founder of Grow Your Oyster Reefs. And she will be sharing about her work using biomimicry to help restore coral reefs. So keep an eye out for our Reef and Brief newsletter. Once again, if you're not on that mailing list, just pop into the chat, click that link, and send us your email. We won't spam you, we promise. We send it out once a month with news and other updates, and the registration for our monthly speaker series. And again, if you're looking for a unique gift for mom, or if you're like me and you just love chocolate and you need an excuse to take a VIP tour of a chocolate factory, please consider going this weekend, this Sunday. 100% of the proceeds for the tour will be donated to MNMRC, will go to help protecting these precious coral reefs that we just got through talking about. So mahalo so much for your support. We hope you enjoy it. And if you'd like more information, or if you'd like to enroll in our newly launched Reef Friendly Landscaping course, you can use the link in the chat. Or if you have any questions, please email rfl@mauireefs.org for more information. And of course, as always, a recording of this talk will be posted to our YouTube channel in a few days. But until then, you can follow us on Instagram or on Facebook. And actually, this was live streamed to Facebook. So if you just can't wait, it should be there right now or shortly after we end this talk. And a big mahalo to all of our wonderful supporters, our partners, our sponsors, just so many. What we love so much about this organization is there's just so many people who just want to be a part. so many community members, so many businesses. So thank you guys for being there, for making us what we are, and we look forward to seeing you guys all next month. So until then, please take care, and we will see you all next time. Have a wonderful evening. Aloha ahui ho.